So now we're going to talk about gene set analysis. And this is a step that you do, or you can do, after you've done your differential gene expression analysis. And so what we're trying to see here is once you have, especially if you have a long list of genes that are differentially expressed, what might that say about the function of your groups of cells? And so you typically want to compare your list of genes to some sort of database or lists of genes that you know something about. So you might know pathways that have different genes involved. You might know cell types that have different gene markers. And so you want to try to clump your lists of genes together into to understand how much of each of these functions might be affected. So in this lecture, we're focusing on transcriptomics because this is single cell RNA sequencing and differential gene expression, but the principle is the same for other omics data as well. So as I said, we're trying to predict the functional changes of cells based on their expression or differential expression analysis. And an important word here is predict. You can look at lists all you want and find very significant differences, but this will just give you an indication of what might be going wrong and how to interpret your results. But you can't say based on RNA alone that there's definitely a difference in this pathway in like function. And so we do this especially, as I said, to make sense of long lists of DEGs. If you only have a couple differentially expressed genes, you can probably draw some conclusions just by looking up what those genes are about. And then to try to predict what the biological consequences of this these under and overexpression might be. You can do it to try to extrapolate to pathway activity. And also an important part of this type of gene set analysis is that rather than looking at one gene at a time, if you see a smaller difference in a lot of genes from one function, that might be more telling than having one gene from one function where you have a large difference. And in that way, it's a bit like it's not as sensitive to false positive differential genes because it'll be a bit more like it might have some false positive and some false negative, but overall from the whole pathway, it will be a bit more reliable. So in order to do this, you need to have your differential expression results. You need to have gene sets, which are lists of genes. You can do this on one gene set, but typically you want to do it on multiple. And then you need to have some sort of statistical test. Where to get these gene sets? Typically, you will get it from databases that have been curated in some way. Gene ontology is a very popular one. KEGG is a popular one. Reactome and MSigDB are also quite well used. But it doesn't have to be a list that has been curated by a whole community. It can also be based on previous studies. If you have something that like where you have a differential expression analysis from something else, you can see how they overlap. If you have markers of a certain population that you have found, you can see if your genes are enriched for that. And also, if you have a transcription factor of interest where you have compiled a list of targets, you can also look at that. As for the databases, gene ontology, as I said, is one of the most popular. It's a network of terms, essentially. So each of these terms is one list of genes. It's hierarchical in the sense that they'll be grouped according to, for example, Hexo's biosynthetic process is a type of Hexo's metabolic process. And so there will be a lot of, this will have a subset of the genes that are in here. And this can mean that you get a lot of results from very similar gene expression terms, which is a bit of it's a bit of work to then go through it and see what is actually of interest for you. There are three types of terms, so three ontologies. There's the biological process, which is very much like about what these genes cause the whole cell to do. So like neutrophil chemotaxis, cell proliferation. And then there's more molecular function of the protein, the product, like histone acetylation or phosphorylation. And then there's the cellular compartment for where this protein is in the cell. And genes can, of course, belong to multiple terms. In addition to, in the hierarchical sense, genes have multiple functions and functions overlap. So you can get a lot of results even from a small list of genes. Then there's also KEGG, which is smaller. There are fewer terms in there, but it is highly curated, and you also get some sort of pathway showing how the genes interact with, or like how the proteins affect each other. So once you have your list of genes, like your differentially expressed genes and your gene lists of interest, then you need to apply a statistical test. And one of the most common ones is overrepresentation analysis. It's a very simple test in the sense that it's just looking at in your list of genes and in all of the list of genes, or in all of the genes, for each gene set in this database or whatever, how many of those genes are in your list and how many are not. And then you can get this type of matrix where you just perform a Fisher's exact test, and then you get a result of whether there's a significant overlap or not. And again, it's statistical significance, so then you actually need to go into the genes to understand if you think this is reasonably going to affect the function. So as I said, you compare it to all of the genes, which is called the universe, or the background, and that can be either all genes ever or all genes that are expressed in your cell population, and this will affect the results. Typically, you want to have some reduction so that you're not just getting that these genes are expressed in this population, because if you're just comparing that population, you already know that that's the case. So if you're just comparing the genes that are expressed in any of your cells, then you get a more exact estimation of the difference. It does require an arbitrary cutoff. So you have to say, these are the genes I consider significant, and these are the ones I don't consider significant. And it also removes any other information. It doesn't consider the ones with a very low p-value more important than the ones with, p is 0.04. If you set the level at 0.05, and it also doesn't consider the ones with a higher fold change more important than the ones with a lower. You can also, I forgot to add this also, but it's also important to consider whether you're looking at, like in your list of DE genes, are you including all differentially expressed genes or just the ones that are positively like the ones that are overexpressed, because that affects your interpretation of what is going on with the pathway. But then it's also important to remember that a pathway can include both inhibitory genes and activating genes. So, then the other most common test to use is the gene set enrichment analysis, the GSEA. In this case, you don't set any threshold for which p-values you consider significant. You just order your genes based on significance, and typically this will be based on log fold change. And so you can see here that when it's ranked, you can see generally that these genes are the ones that are more overexpressed in group A, and these ones are more overexpressed in group B. And then you have all your ranked genes, and then you look at a gene set of interest, and you identify where those genes are in this ranked list. So for example, in this case, each line is one of those genes, and then you can perform this random walk test where you look at for each time that the gene appears in your ranked list, it takes up one, and that way you'll see if your genes are overrepresented in either of the ends, or if it's just a homogeneous distribution. And this also considers the extent of the log-fold change, not just the rank. And then you get this enrichment score that tells you how uneven the distribution is, essentially, and so how strong the enrichment is. And this is the type of plot you'll usually get if you use the original GSEA tool. So you have this walk, and then you have the where the genes are, and then the log-fold changes of your genes. You also have the normalized enrichment score, and which is a modified enrichment score. And then the advantage of this method is that you don't need to have a cutoff. You don't need to say that these genes are significant and these aren't. And it does take both the order and also the extent of the difference into account. So genes with a very high log-fold change will be considered more important than the ones with a bit lower. And so this one can be a bit more sensitive to subtle changes. My main issue with GSEA is that you often have to choose whether you want to look at p-value or log-fold change. If you have a big population, that is typically not a problem. But if you have a population where you have outliers that are shifting the mean so that the log-fold change is high, even when the p-value isn't significant, that can affect this analysis. But yeah, so those are the main two options. And then as to how to apply them, there are online tools where you can just paste your list of genes, and they will look at a lot of different databases for you. Actually, Gorilla might just be gene ontology, but Enricher looks at many different ones. And then you also have tools that you can use in R or Python. For example, this is a GSEA function. This is one that combines different, this is more of a wrapper function, or wrapper package, so that you can apply different tests on different databases. So things to consider when you're using databases, especially, there is a bias in curation. So highly researched topics will be overrepresented. Typically, you'll get a lot of immunology, enriched data sets, because there's a lot of description of immunology and cancer as well. Whereas if your topic is a bit less research, you might not get those results just because those gene sets don't exist in the databases. Gene set names can be misleading. For example, you might be getting that in your top enriched gene sets, you have COVID or prostate cancer or something like that, and it's really just immune genes or cancer genes. So it can be unnecessarily specific. So you need to check whether it makes sense to say something about that disease specifically. And that is also related to specific versus general gene sets, that they might be overly specific or they might be overly general, and you might find something more interesting if you look at the more specific. Yes, the question?
Yeah, I had a question relating with that it gives you these very broad terms like immune response.
Mm-hmm.
And then in that case, would you go manually check the genes in literature or with collaborators or something? Or is there any other tool that you can dig into more specific?
Yeah, so one thing you can do is to look at if you have multiple terms, is there an overlap in the genes between terms? And then you might see that all of the ones that are in immune response are also in neutrophil chemotaxis. So it's probably that one that is the important one. So that is one way, and it typically won't be all of them, but it can give you an idea of where those main differences are. I can say that in general, showing that there's an immune response enrichment is probably not super interesting, especially if you're working on something where you expect to find it. It can be like a good sanity check, but it's probably not something that you want to present as like, look at this novel result. But yes, like when it comes to then looking into the genes, I don't know really of, other than like comparing to other data sets, it is kind of having to go into individual genes and look at them. Sometimes it'll be like one family of genes, which, then it's easy to look up what they're doing, but it also affects your interpretation because it might just be that there are a lot of genes kind of doing the same thing. Yeah, I don't know if I have any other tips on evaluating, but also another part of evaluating is also looking at how, like in those genes, what are the differences actually like between your populations? So that is also something to consider. You also need to consider multifunctional genes that can show up in pathways where they might not have an impact in your cell type or something like that. Also, different databases can be using different gene IDs, like in terms of some use ensemble, some use entree, and things like that. And whenever you're doing a translation, that can be affected by, what the translation is based on, like which database you're getting the translation from. And also databases can change. So sometimes if you rerun an enrichment later, you might get a different result. And that's just good to be aware of. Curation is organism specific, and also, to some extent, it can be based on as a different cell types, which might affect the interpretation. But when it comes to organism specific ones, you can sometimes look at homologs, but it is better if you have a gene list that is based on your organism, so that you know that those genes actually have that function in that organism and isn't just a homolog to a gene that has that function in another organism. And critical evaluation is required. So all of these things to consider, and then also going back to your cell level expression data, and to always try to use this as a starting point for validation. There's just one reference in this one, but I just want to acknowledge Leif Vigge, Paulo Starnowski, and Roy Francis, who had this lecture before.